Today I have the privilege of interviewing Dr. David Harper. David is the lead editor of a textbook on bacteriophages that Springer Publishers is releasing this summer and has carried out groundbreaking research in this field as the CEO of biotech pioneer Biocontrol and more recently as the chief scientist of Amplify Biosciences. Welcome, David. You are currently a member of a panel called Alternatives to Antibiotics, which is a collaboration between the Wellcome Trust and the UK Department of Health. Infectious disease and the growing problem of resistance to antibiotics seems to be one of the main focus areas of the group. What are some of the technologies being looked at?
Well, it won't surprise you if I say bacteriophages and their various manifestations. The main problem with that is having a publication coming out very soon in Lancet Infectious Diseases. In fact, it was submitted in its final form today. I can't say too much about it, except to say that it's a very broad review of alternative technologies, from biological such as phages through variant forms of small molecule drugs, addressing new approaches, disrupting various aspects of the bacterial metabolism and the bacterial colonization process. What I can say is that it's a very broad review with lots of people from both industry and academia. And the report should be up fairly soon. The conclusions broadly are that there are a lot of technologies out there, but we need to put the resources into developing them. Because if we don't, we're going to hit a very real gap between the availability of antibiotics via the conventional approach and the availability of antibiotics via any new approach. And there are recommendations on funding. Whether they get followed up, of course, is another matter entirely.
The UK Parliament's Science and Technology Committee stated recently that antimicrobial resistance has the potential to send medicine back to the early 20th century. They believe the only way to ensure effective treatment of constantly evolving bacterial pathogens is to continually find new antibiotics. Do you agree with this statement, or are there alternatives out there that can prevent bacteria from adapting and developing resistance?
Well, that is something that the panel has looked at, which is ways to inhibit the development of resistance, ways to inhibit the manifestations of resistance. You don't have to go far to find that. There are quite a few combination drugs already on the market where you pair an antibiotic with the inhibitor of resistance. And that approach has real potential to extend the life of conventional antibiotics, even with something as different as bacteriophages. Some of Amplify's work has shown and published that you get a synergistic effect, that the bacteriophages do help antibiotics to work where they previously didn't. So I think any technology can be combined, particularly when that technology has a completely different base, so you don't get cross-resistance. You have fruitful ground for expecting synergy, or at least combination, and you do seem to get that.
Okay, the pipeline of drugs to combat infectious disease is not nearly as robust as it was years ago, and the Wellcome Trust believes this is due to the low price tag of such drugs compared to the more lucrative candidates such as cancer medications. And also the fact that new antibiotics are rarely used as a first-line defense, along with, of course, the high cost of the FDA approval process. Do the recent legislative changes in the U.S. provide companies with enough of an incentive?
You want the short answer? No. It is worth small companies doing this. For example, I'm not giving anything away because Amplify have said it in public. But billion-dollar markets are easily predictable for antibiotics which are used even to combat drug-resistant infections from the escape pathogens. So there is a market there, but whether that market is big enough to pull a big company away from the next statins or the next Viagra, I don't know. There's enough there to justify a small company, but when that small company tries to sell to a big company, they run into the same problem. So I think you need to do, not you personally, more to support the necessary investment, not only at the basic R&D stage, which is carried out by the small companies, but also to encourage the big pharma companies to partner with the small R&D companies, because if small R&D companies put it all into getting the thing through phase 2 and then sit there going, hey, we're clever, look at us, come and buy us, and big pharma goes, no thanks, then it's all for nothing.
Well, yeah, that can be a little bit discouraging, I would imagine. Can you give us a quick background in layman's terms on the history of bacteriophage therapy, how it works, how it addresses the growing problem of antibiotic resistance, and some of the potential applications?
Certainly. Bacteriophages were first discovered precisely 100 years ago by an Englishman called Frederick Taut. He didn't really follow up on them, being somewhat distracted by being sent to fight in the First World War. It was then followed up on by a French-Canadian called Felix de Rauw, and he really did follow up on it, in fact coining the name bacteriophage. And within a couple of years of his observations, he was trying to use them in therapeutic approaches with apparent success. Wouldn't meet current FDA standards. For example, the safety testing consisted of the investigative team drinking the bacteriophage preparation. Can't get away with that these days. But there was apparent success through the 20s. The problem was nobody really knew what phages were. They weren't known to be anything. In fact, I've got articles in the 1940s saying that it's been conclusively shown to be an enzyme, which it isn't. It's a live virus which infects bacteria. So nobody knew what they were. Nobody knew how to make them efficiently. Nobody knew how to make them properly. A lot of the early preparations were completely inactive, at least reported, and I'm still trying to track that one down. And basically, they were promised to work on a variety of things. I have a slide presentation on this, promising that bacteriophages can cure urticaria, eczema, and herpes, which they can't, frankly. And the science wasn't there, so they were used improperly. They were used incorrectly. And that really, when antibiotics came along, demolished their approach. When the only alternative was salvarsan, an arsenic compound, then bacteriophages made sense. When penicillin and some of the early antibiotics came along, they made less sense. So they were marginalised, pushed out to mainly Eastern Europe, where they continued to be used. And it was only when the antibiotic crisis became really apparent in the 90s that attention switched back, finding some work from the 1980s, which was done to very high standards, which showed they apparently worked in animal systems. And really, all of the interest in bacteriophages revived from that work by an Englishman called Willie Smith and his assistant, Bill Huggins, and went from there. And that's where Biocontrol and then Amplify came from. It's where the other companies, such as Pharmaceutics Pharma, Belgian Army, where they came from. And what they're working on is really all based on that, as much as it is on the essentially anecdotal evidence from Eastern Europe. And as you know, there have been a number of safety trials, but there's only been one reported efficacy trial to date. That was one carried out by Biocontrol in 2006-07.
You're beating me to the punch there. That was going to be my next question. So definitely tell us about your trial. Just to fill people in, Biocontrol carried out a phase 1 to 2 trial on chronic ear infections by Pseudomonas bacteria. So what did you find out then when you did that study?
Well, just to put it in context, the reason we picked Pseudomonas is there are a number of infections where Pseudomonas is the problem. Bacteriophages are very specific. You can't give a say to somebody, you've got a bacterial infection, have some phages. That was what they did in the 20s, and it doesn't work. You have to know what the bacteria you're dealing with, because almost all bacteriophages are very species specific. In fact, they only work on some strains within a species. You generally have to use a mixture to get broad coverage, although there are exceptions. So we picked Pseudomonas because chronic late-stage ear infections are pretty much pure Pseudomonas. Cystic fibrosis lung, which Amplify is working on, is pretty much pure Pseudomonas. Burn infections, which Pharmaceutics Pharma are working on in the trial that's currently running in Europe, they are Pseudomonas infections, and you can use a specific agent there. So we picked the chronic ear, partly because there is actually a similar condition in dogs, and we developed in parallel a veterinary drug. And that was trialled for pet dogs, and you never know, may well come back for pet dogs who get chronic, horrible Pseudomonas ear infections, and having your doggy drip green pus all over the carpet isn't very popular with owners, or indeed I imagine with the dogs. So we trialled a phage mix there, and then we trialled a phage mix in a very similar infection in humans. And, well, it's reported in Wright et al. 2009, we got very positive signs. It was a very small trial, it was necessarily limited. It was the first trial ever to look at the efficacy of phages to modern standards, and yeah, it was a success. So the outcomes of that were extremely promising. And that really has been largely responsible for fueling the continued interest within biocontrol and Amplify, and done quite a lot for the sector as a whole. I mean, a lot of people feel that it was a very small contribution and that the evidence from Eastern Europe is overwhelming by comparison. And certainly the evidence from Eastern Europe is extensive. The problem is that the regulatory agencies have a fairly negative attitude towards incorporating that evidence in any submission, and it is regarded as anecdotal. So you need the hard trial evidence, and Amplify Biocontrol, as they were then, are the first ones to have that.
Yeah, I understand a lot of these Eastern European trials often didn't conduct double-blind studies. They didn't really follow Western standards as far as having a control group and some of the other things that are kind of expected when you run an FDA trial.
It's not just Eastern Europe. There were trials in India in the 1920s, the bacteriophage experiment run by Lieutenant Colonel Morrison with one arm, and the reporting on that was of such a standard that, in fact, I think it's about 1.5 times the population of the area which was where the trial was based appeared to have been treated.
No kidding.
So you can see that there are a few flaws with that one. And yes, one of the biggest problems with the Eastern European trials is they weren't reported to Western standards. One quite forward group, who I won't name, reported success on the basis patient didn't feel better, patient felt better, patient felt much better.
Yeah.
And that's not going to be up to FDA standards, I'm afraid, no matter how you repackage it. And a lot of the Eastern European trials were reported in the very broadest sense. As you correctly say, control groups were often absent or somewhat compromised. One trial treated one side of the street and left the other side of the street alone, for example. And you just don't have the level of reporting required to make it usable by Western agencies now. It's a pity, but it's a fact.
Yeah, I understand also that they had some issues with finding the correct dosage for the bacteriophages, that there were some trials where the results maybe could have been a lot better if the dosage had been different. But
I'm going to disagree with you there, I'm afraid. Dosage isn't really an issue with bacteriophages. The reason being that they replicate. They're different to every other antibiotic approach. They are functionally an antibiotic, but they're not a conventional antibiotic. When they get in, they replicate. Thus, the dose you give initially is merely the spark that starts the fire. If you want to give enough bacteriophage to kill everything first off, as some approaches do try, non-replicating phage approaches, you have big issues with the amount of phage you have to give, with the amount of phage which actually manages to penetrate through barriers. Phages are quite big, they're sort of gigadalton range lumps of nuclear protein. So, for example, to give enough bacteriophages to match the number of molecules of penicillin in a standard therapeutic dose, you would have to give somewhere between 300 kilos and a ton of bacteriophage. So, relying on the input dose is not going to do it. To my mind, although some people disagree with me, it has to replicate. And that replication is the key. It's the same with live vaccines. The dose of chickenpox vaccine that you give is 400 live viruses. If you heat that and kill it and give it to somebody, nothing happens. If it's live, it replicates, it grows, and you get a stimulating immune response. It's the same with phages. They're even safer because they can't infect our cells. So the dose you give can be very small. In our human trial, the dose was 5.4 nanograms. That's published, I can talk about that, even though I've left Amplify. And 5.4 nanograms is a billionth the dose of a conventional antibiotic. But because it replicates thousands or millions of times, depending on where it finds its lunch, it can make enough to be effective, even from that tiny dose. In one reported trial, which was done by the chap who Biocontrol licensed the technology in for, the dose was actually picogram range. And overnight, the dog, it was the dog's ear that was being treated there, the vet came along the next day, he lifted up the dog's ear, a big floppy St. Bernard, he looked, he said, wrong ear. Went round to the other side, no, that was the right ear. Overnight, massive clinical improvement. From, again, it was, I think, 400, a few hundred anyway, active bacteriophage, and that is a picogram range dose, because they multiply.
That's pretty incredible.
Yeah.
Okay, let me move on to one of the commercial concerns about bacteriophages. James Anderson, the director of European partnerships at GlaxoSmithKline, has said that the use of bacteriophages hasn't received the level of investment it warrants because of the challenges around securing intellectual property. Phages are a naturally occurring substance, and like you point out, they've been used since around World War I. So, what are some of the ways companies can fortify their patents so the considerable expense of getting a drug candidate through clinical trials is not wasted?
Well, antibiotics have been used since World War II, and we patent them. Phages are new. The phages which go into any product are new and are new biological entities. But the best analogy is monoclonal antibodies. Because of a rather strange decision by the British Medical Research Council, the basic technology of monoclonal antibodies was never patented. So what you have there is a massive number of biological entities. You can make more in a couple of weeks very easily. You have no patent protection on the basic technology, and yet monoclonal antibody drugs are worth several tens of billions a year. Because when you develop something through the whole clinical trials process, anybody who starts out and says, hey, I can make one of those, is starting 5, 10, 15 years behind you. And what's the life of a patent? So I don't see patenting as absolutely critical. You can protect things simply by being first to market, as happens with a lot of monoclonal antibodies. You can make another antibody to respiratory syncytial virus, and yet the existing product there has a market value. Just because you make another one doesn't mean you've got it to market. Added to which, it is possible to patent bacteriophages. I know this because I've done it, as have other companies. It is possible to protect them, and the recent Myriad interpretation says you can't patent naturally occurring substances. You're absolutely right. But following responses from a variety of sources, mixtures are patentable, and very few people are planning to use a single phage. Micrios's product, Listex P100 does, but most products will be mixtures of phages, and those should be patentable. Now, there is the issue of how do you stop somebody from taking your mixture, growing it up in some bacteria and saying, hey, I've got a new mixture. Well, first, they would have to go through clinical trials if they claim it's different. If they're not, you got them. Secondly, there are ways, I can't really say anything because it's an amplify issue, which I was responsible for and spent considerable time on, but there are ways to protect derivatives. So I would have to disagree.
So I think what you're referring to there is I've seen in some of Amplify's reports where they'll take, for example, mutant strains of bacteriophages and put a patent on that. And then I understand there's a company called Synthetic Genomics that's actually genetically altering bacteriophages. And I would assume that that would also be another thing that could be done to enhance the viability of your patents?
You can certainly do that, yes. If you make a sequence which is entirely novel, then it's patentable. Full stop, the end. The issues are, firstly, somebody can come along and make a few tweaks. If you've got a CGTCCCGA patent, somebody can alter a couple of the non-essential areas and say, well, it's a new sequence, mine. Second, getting anything genetically engineered into the clinic is challenging. Going to the regulators and saying, here's a new antibiotic approach. It's self-replicating, it copies itself, it makes millions of copies. Yeah, and they do seem receptive to that. By the way, it's also genetically engineered, is a little bit of a step too far. Actually getting into the clinic with something which is genetically engineered has a whole host of additional challenges. Synthetic Genomics are doing it. I know of at least 2 other companies which are doing it. But to give you a little story, a company called Parecetes Pharma set up in France to work with genetically engineered bacteria. First popped up in 2008. In 2013, stood up at a conference and said, we accept that natural phages have to be the first to market. And they're now using natural phages in the Phagoburn trial in Europe. So people have backtracked from the idea of GM, except those people who are entirely GM in nature, like synthetic, who I have a lot of respect for, and I know people in synthetic. But you have to be aware of the challenges in getting those GM phages to market. There are certain areas where the natural phages are unsuitable. I can name a couple of targets, but I won't do, I'm afraid. where the natural phages have issues and where genetic engineering will be an easy way around that, were it not for the fact that you've then got to go to the regulators and say, hello, here's my new GM product. And they say, good, go and talk to those 7 committees over there.
Interesting. Okay, I didn't realize that there are obviously some serious drawbacks to that approach. Also, it
Well,
seemed to me like a no-lose situation there, but obviously it sounds like getting it through the FDA approval process can be a whole other story.
I should say I've never tried to get a GM phage through the FDA approval process simply because the perceived barriers are so great. For example, Scotland has just banned GM crops. You know, there is this negative perception. And if you can get away with that, if you can get a GM phage through, good luck to you.
Um, Let me move on here. I understand one of your main interests is in the use of bacteriophages as a biological control. What can you tell us about that?
Well, bacteriophages are a biological control. I actually have used graphics from classic agricultural biological control when explaining how phage therapy works. They go in, they replicate, they spread, they wipe out your target. That is biological control. So the PKPD, the kinetics, are completely different to a conventional antibiotic or even to a non-replicating protein like a lysine. They do work like a biological control, and they're not at their best when they're wiping out the very last few of their target. For example, snowshoe hare and lynx, the classic predator-prey relationship. If you almost run out of hares, the lynx starve, and then the hares regenerate, and the lynx follow them. The classic predator-prey cycle, with the predator lagging behind the prey. And that's the way phages are. You have to use them in a way that acknowledges they are a biological control with the kinetics of normal biological controls. A lot of people make the mistake of thinking, hey, they have an antibiotic effect, they're an antibiotic, let's use them like an antibiotic. No, you can't, sorry. There is a chapter in the book, I know that, so I'm writing that chapter, on phages as biological controls. And more broadly, my interest came about, as you would guess from the name of the company, from biocontrol, biological control of a range of things. Our interest back then was broader than bacteriophages, but they focused in on bacteriophages. I've now left Amplify and I'm working on a broader application of biological control, taking it into areas of biomedical importance. It's been ghettoized in agriculture, where the margins are small and where the challenges are high. If you use a live virus, for example, on a tropical coconut plantation, as is done, it's inactivated by ultraviolet light. You have to spray at dusk. It's washed off by rain. It can get blown away by the wind. Whereas if you use it inside somebody's mattress, which is a nice constant environment, to go after their dust mites, which is what I'm doing now, then it is a very practical approach. And the biggie, as I said on BBC Horizon a few years ago, I nearly had this ready to play for you, the biggest problem with bacteriophages as a therapeutic is nobody has yet developed one. And that is the big issue. There is no clear path to market. But biopesticides, there is very much a clear path to market. The US Environmental Protection Agency's website, which I've quoted repeatedly, says because biological pesticides are generally safer and more specific, the actual amount of evidence that's required to get them to market is less. They are faster to market. So skipping out of bacteriophage therapy, oh dear, can we get this thing through the regulatory process? Into biopesticides, yes, of course they can go through regulation. And the cost of field trials is considerably less than the cost of clinical trials. Strikes me as a damn good idea. So that's where I am now at my new company, Evolution Biotechnologies.
How big would you say the market opportunity is that you're shooting for?
€1 billion. That's in primary markets alone, EU and US. Expand out of that to the other main areas where there's a big dust mite problem: Japan, South Korea, India. Middle classes of India are very much able to spend money on things like this. South Africa, huge death rate there. And even some dry countries, one of the highest death rates among the developed countries is in Qatar in the Gulf. Because when somebody makes the inside of their house comfy for them, with humidifiers, with carpets, they make it comfy for dust mites. Then it's a €1 billion market in the US and EU, another €1 billion in the secondary market. It's big.
So are you planning on focusing on human therapeutic applications, or are you also looking at agricultural applications?
No. Biomedical. Biological control is still expanding at about 8 to 10% a year in the agricultural sector, but it's not being worked on in the human biomedical sector to anything like the same extent. And it's pretty obvious how to do it. You combine what I've done with bacteriophages, where I know how to get them to clinical trials, and what's done with agricultural biopesticides, and you have biomedical biocontrol. And that is, I think, an area that is ripe for expansion. That's why we recently filed a patent on our lead product, the dust mite control agents, and that's why I can talk to you about them. And I think that's got a huge potential.
And it sounds to me like most of the applications would be in developing countries as opposed to, say, the US and Europe.
New. Asthma is a disease in the developed world. The primary trigger is the house dust mite. That is a developed world disease. Second target, bed bugs. Where are they a big problem? New York and American hotels across the country. I've been bitten at in New York, and I didn't enjoy the experience. If a hotel could say, we are bioprotected against bed bugs, that would be a plus for me, and I think it would be a plus for a lot of hotel chains. Cockroaches, they're another trigger for asthma. Interesting, there's hundreds of cockroach species, but there are only 3 which are really, really widespread. A couple of dust mite species, 1 or 2 bedbug species, 3 cockroach species, biological control specific. Where you can find just a few targets, biological control has a real plus. It can spread. You don't have to treat every surface. You don't have to, for example, with dust mites, you can get your sofa taken away and put in an oxygen-free environment for 2 weeks. That will kill the dust mites. You can put it in the freezer, but it's a bit tricky. Put the sofa back, of course. The first thing that happens is the dust mites in the carpet go and run up the legs. There's a tale of bed bugs. A chap cleans hotels, and he just finished cleaning this hotel. He has sniffer dogs that detect bed bugs. And he just finished cleaning this hotel. He was walking the dog out through reception, and somebody had a trundly suitcase, and the dog went, "Wow, whoa, 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 whoa." Suitcase full of bed bugs. Recolonisation. Biological control takes care of that. There's a graph I use in my publicity of the company, which shows treatment of a cucumber crop in Finland with a biological control against the chemical control. The chemical up, down, up, down, up, down, up, treat, down, up, treat, down. The biological, 1 dose, beginning of the year, flatline. And that's what you can achieve. The biological control can spread. It can get from the carpet into the mattress, or the mattress into the carpet, into the soft toys, which are full of dust mites, into the pillows. A 6-year-old pillow is about 10% dust mite material, by which I mean mostly dead dust mites and dust mite poo. And you can get into all of those environments with biological. No existing treatment can treat those areas together. You can bury your mattress, you can spray your surfaces, you can put a caricide in, mite-killing chemicals, and they'll work wherever they get to, but they won't spread. A biological can.
So it sounds like a lot of these applications are environmental applications where it wouldn't be necessary to go through the FDA approval process, and we're talking literally about spraying, say, a mattress as opposed to a patient actually taking a medication?
Yeah. You'd have to be happy that the material is safe to use in the same environment as an asthmatic patient, obviously. But the field trials are simple physical field trials and are cheaper by an order of magnitude than clinical trials. And there is an improved path through all that process. The way, for example, the dust mite product will probably go is committee stage COPR in the UK and EU, and then potentially GRAS into the United States as food products with bacteriophages have been approved. If you have something which is generally recognised as safe and is an environmental agent rather than a clinical agent, there is the potential for GRAS. Now, this remains to be proved, obviously, but it will mean that the whole process could be considerably simpler than the approval of the therapeutic.
My final question is a hypothetical one, David. If you could own shares in just 5 clinical stage publicly listed biotechs, which ones would you choose, and what is it you like about their pipeline or their drug platform?
I've stretched that question very slightly. I've got my answers here. Going with the sort of conventional antibiotic approaches, I think Sempra and Tetraphase are both quite interesting. They've both got interesting pipelines. They're both phase 3 or past phase 3, and I think that's important. I should say that a lot of the companies I know that have interesting pipelines are preclinical, are early stage clinical, and those companies tend not yet to be public. There are some companies with quite advanced approaches which aren't yet public, so I can't put them in this category. So I'm a bit restricted. For example, in the whole phage sector, there's only one public company, and I just left it. So I think those two, although it's conventional antibiotics, they're quite interesting. Going a bit closer to home, although defining them as clinical stages less well defined, Contrafect, a company which I know from seeing your writings you're a bit of an enthusiast for. And again, they've got some very interesting approaches. So, Sempra, tetraphase, Contrafect for sure. And then I'm afraid I'm going to shell out a bit on the last two and go with the always have a percentage in blue chips and say GSK and Merck. GSK are very active in antibiotics, unlike a lot of other companies in the area, and Merck have acquired some very interesting approaches, including vaccines. So that will be my 5.
Appreciate it, David. Thank you so much for spending the time with me today. Wish you the best of luck with your new venture, and look forward to the contributions your company Evolution will make to combat this growing healthcare problem.
Thank you. It's been a pleasure talking to you.